This particular session, we will be discussing different pathogenic microbes. To start with, I shall be discussing today the role of Staphylococcus aureus, that is their pathogenicity, development, characters, everything. Now this particular bacteria, Staphylococcus aureus, is round or cocci which remain in aggregate, and when seen under microscope, as you can see here, they are gram positive, so they are blue violet in color, and they are just representing an aggregate of grapes. This particular bacteria actually belong to the phylum Firmicutes. It is having the class of Bacilli and the order is Bacillialis. It is belonging to family Staphylococci and the genus is Staphylococcus, and of course the species is Staphylococcus aureus. This particular bacteria was discovered way back in the 1880 to be specific, and it was discovered by the surgeon Ogstone, Alexander Ogstone. When he was operating a patient in the knee abscess, he found out some pus cells, and in those pus cells there were this Staphylococcus aureus bacteria. And now of course we can see their electron microscopic structure, and they clearly represent an aggregate of round grape-like structure. This particular bacteria can remain anywhere in human body. It can remain in the skin, it can remain in the soft tissue, it can remain in the glands, even it can remain in the heart. As you can see, it will be colonizing in the valves of the heart, in the wall or the endocardium of the heart, causing the dreadful disease of endocarditis. When they are grown in normal nutrient medium, they are actually represented by orange colonies. And when they are grown in blood agar plate, it was observed that they can actually degrade hemoglobin. So they have this property of hemolysis, and this is where they are producing this colony, and they have actually discolored the hemoglobin. They have different properties by which they express their virulence. This virulence can be in terms of the production of certain enzymes. One of these enzymes is catalase, and this catalase actually they will remove the reactive oxygen species, and thereby they will make the bacteria colonize without causing any harm. They also, when grown in this particular triptych soya agar, they will produce a unique pigment, and this pigment is the golden brown pigment which is called staphylozanthine.
So does staphylozanthine have any role in the physiological activity of the bacteria?
You see, staphylozanthine apparently is a carotenoid pigment. So it is obvious we can think that this particular pigment is involved in photosynthesis. But this bacteria is non-photosynthetic. So staphylozanthine as such is not having any role in photosynthesis. Then what is the way? Actually, this is produced from farnesyl diphosphate, which is also producing cholesterol. So cholesterol and staphylozanthine, they definitely indicate they are producing the cell wall component of the bacteria. And that is the reason why when we take some cholesterol inhibiting drug like statin, this particular bacteria can be checked apart from other antibiotics. Now, they also have the unique ability to produce enzymes like coagulase and kinase. Now, what do? This coagulase, you can see, they will actually initiate coagulation of blood, and thereby blood clotting will be inside the blood vessel, and that can be harmful. But after the coagulation, they also can remove the clog by the enzyme kinase. So that is another property by which they will coagulate at certain portion of the body, and they will remove that by kinase action. At the same time, they can also produce an enzyme called hyaluronidase. This particular enzyme will act on the substrate which is present in our tissue called hyaluronic acid. And you know, hyaluronic acid is a polysaccharide, and this polysaccharide contains the units of D-glucuronic acid and acetyl D-glucosamine, so they will break that. And thereby, when they break that, this particular bacteria can be released from a cell inside, and they come out and they invade the blood. At the same time, they can also produce an enzyme called DNase. It's obvious because they will attack the nucleus and they will break the DNA. And when they break the DNA, this is how the DNA nucleotide is there. There's the phosphoric acid, pentose sugar, and the nitrogen base. So that will be broken down to oligonucleotide. So the DNA can be directly broken by this particular bacteria. with the enzyme DNase. They also can produce an enzyme called staphylokinase. Now this particular staphylokinase actually is a very important enzyme which will digest protein like fibrin. So at the same time, when we have a clot, we have a fibrin protein. So this clot is broken, and thereby, whenever there is a clot, the bacteria cannot move from one portion to the other portion. But if they have the property of producing this type of staphylokinase, they will go out of this clot and they will invade newer areas. So it is also harmful for us because it is spreading in our body. Of course, this is very important. They are resistant to antibiotics. Now when we talk about antibiotic, the most important antibiotic is penicillin group of antibiotics, and they are having this particular beta-lactam ring. So they will produce an enzyme called beta-lactamase or penicillinase, by which penicillin is converted to penicilloic acid, and when it is converted to penicilloic acid, the penicillin is of no use. So this is how, when we apply antibiotic, this particular staphylococcus will become resistant to it. Apart from producing different enzyme, the bacteria can also produce toxin. And these are very important tool to invade the host cell. One such toxin is the enterotoxin type B. And this particular toxin will be produced whenever they come in contact with the host cell or they are trying to invade the host cell. One such particular action can be seen in the tampon-mediated infection of Staphylococcus aureus. What happens is, this particular bacteria present in the tampon will be actually getting inside the vaginal endothelium, and they will actually incorporate the production of different T cell. And this particular toxin, which is actually acting as an exotoxin, will enter into the host cell and will activate the antigen presenting cell, producing different types of interleukin, causing hypersensitivity. And this particular antigen is called the super antigen.
Sir, I have a question.
Yeah.
So why it is called super antigen?
You see, this particular normal antigen will bind with a specific receptor and will enter and will have the agglutination reaction. But this particular super antigen will enter into the host cell through the MHC2 complex without coming in contact. And at the same time, it will invade large number of such cells and will cause massive production of interleukin, causing hypersensitivity. So that is why it is called a super antigen or a powerful antigen, you can say. Okay. Apart from that, this particular bacteria also produces another toxin, which is also a protein though, called exfoliative toxin. And this particular toxin is mostly active on the skin surface. What happens is, this particular Staphylococcus colonization in the skin will produce a unique syndrome called SSSS or Staphylococcus scaldin skin syndrome. What happens? This is the stratum corneum, this is the stratum granulosum, then the stratum spinosum and the stratum basal. What you can see, whenever the bacterial colonization is here, the protein linkage in between the skin cells will be degraded by the bacteria. So what happens is, these particular skin cells present in the stratum corneum will come out as scales. So that is why it is called the Staphylococcus scaldin skin syndrome. And it is brought about by this toxin under the action of this particular exfoliative toxin. There is another very important role of this bacteria producing what is called pantene-valentine leukocidine or PVL. And this particular protein will actually bind with protein A and they will activate the TNF receptor and they will bring about different neutrophils towards this particular reaction center, causing massive chemokine production, which is also similar to producing hypersensitivity in the host cell. And this particular PVL or this pantene-valentine leukocidine actually was not originally in the bacteria, it was originating in bacteriophage. And this particular protein A, which is actually acting as a receptor for PVL, is binding with the PVL and causing the massive change. Apart from that, this particular protein A, which is also present in the particular bacterial cell, will also cause immunosuppression or immunosuppressive activity. So that way the immunity is reduced in the host body.
So I have a question.
Yeah.
So is there any positive role of protein A?
You see, whenever there is a protein which is present in the bacterial cell, this particular protein will actually be able to be targeted by drug molecules. So this particular protein A is a possible target on which we can synthesize newer antibiotics. So every challenge is an opportunity. By utilizing this particular opportunity, we can develop new antibiotics. So what we can see, in order to develop pathogenicity, this particular Staphylococcus aureus has got different role. What are they? They have the protein A, they have the capsule, they have the cell wall, they produce different enzyme, different protein with which they target the host cell and ultimately destroy the host cell. And when this particular bacteria is actually being phagocytosized by the neutrophil, they also can fight with the neutrophil. And this fighting is done by production of different protein and enzyme, which I have already told you. One of such protein is aureolysine, there may be staphylokinase, there may be efflux by which the particular cations are released from this particular body of the bacteria so that they cannot be targeted by the host cell. At the same time, they can break the reactive oxygen species with catalase, and thereby the damage to the bacterial cell can be checked. So you can very well understand that the bacteria is very much protected, or it is having a large number of tools with which they can combat the defensive action of the host cell, and thereby they can actually prevent phagocytosis by the neutrophil. Now we come to the characterization of this particular bacteria. How we can detect that whether it is Staphylococcus aureus or not. Of course, the typical shape, it is just like a grape, and at the same time it is gram positive. At the same time it is producing typical orange colony because of this staphyloxanthin. At the same time, when it is grown in this particular mannitol salt agar, it can utilize mannitol. So that means whenever this mannitol is added, they will be growing, and this discoloration will prove that this is the culture of Staphylococcus aureus. It can produce phosphatase. So when it is grown in phosphate enriched medium, it will also show typical pink colonies, which is also indicating Staphylococcus aureus colony. At the same time, in the present day, the best possible way is to detect by PCR. We call it qPCR. So in qPCR, with a typical nucleotide ranging between 450 to 500, we can say that this is the nucleotide of the Staphylococcus aureus. So perhaps it is the most definitive test to prove Staphylococcus aureus infection. Now coming to the different diseases that this particular bacteria causes. You can see in this figure, it can actually cause infection to different parts of the body. It can cause boils, carbuncles, funicles, pneumonia, emesis. So anything, even it can get into the bone causing osteomyelitis. It can invade the skin causing scalded skin syndrome. It can cause diarrhea. It can cause endocarditis. So you can very well see this particular bacteria is such an important pathogen. It can invade any soft tissue or any organ and can cause havoc. And that is the reason why this bacteria has to be stopped by some means. Not only that, when we talk about human, of course it is harmful, but at the same time it can cause diseases in cattle. This particular capsulated bacteria will colonize, will produce protein channels, and thereby will cause the disease of mastitis in cow. So this particular disease is also important, causing death in cow. But what actually is happening when this particular bacteria is entering the cell? Or I will try to enlist the different intracellular activity that this bacteria is causing. So this particular bacteria is binding with a specific receptor, and this is the integrin receptor. At the same time, there may be fibronectin adhesion. So it can attach to specific receptor, or it can bind with the protein of the cell surface, and it will get inside. Now once it is getting inside, it will be actually getting inside a vacuole. So that particular vacuole, we can call it phagosome. Normally what happens is the lysosome will come in, but the moment the lysosome come in, the bacteria will be released inside the cell. And there may be different types of activity. One important activity is there will be efflux of calcium ion, and thereby this particular bacteria will try to develop hypersensitivity. One such tool is seen here. They will activate the different cytokines, and thereby the hypersensitive reaction will increase. And when this hypersensitivity increases, there will be normal apoptotic function of the cell by which the cell will be destroyed. And one important step in this particular case is macroautophagy, by which the cell is destroyed.
Sir, I have a question.
Yeah.
What is macroautophagy?
You see, whenever the lysosome is causing lysis of the cell, it can actually cause degradation of certain parts, certain organelles of the cell. We call it microautophagy. But here what happens is, massive number of bacteria inside the cell is there. So the lysosome will try to digest the entire cell, and thereby the entire cell will be destroyed. So that phenomenon is called macroautophagy. So macroautophagy is a means by which the infected cell is destroyed. If not, the cell will undergo normal death by the apoptotic mechanism. So the cascade of events that occur after the bacteria has entered the cell and the destruction of cell actually include these molecular phenomena. Okay? Now, definitely, when I talked about that this bacteria is pathogenic, there must be some tools. And these tools actually help the bacteria to survive and also to combat the different antibiotic molecules that you are adding to kill the bacteria. One such antibiotic is definitely the different penicillin group of antibiotics. It can include the vancomycins. Now what happened is, this particular bacteria is having a unique make operon, and this make operon actually can be of different types, and they normally will modify the penicillin binding protein, and this penicillin binding protein is modified, thereby penicillin cannot recognize the bacteria, so they are evading the penicillin attack, and that is true for vancomycin also. Apart from that, they are also targeted by different antibiotics like aminoglycosides. What do? They actually produce different aminoglycoside modifying enzyme. One such modifying enzyme is acyltransferases, then the adenyl transferases, and the phosphotransferases. This particular bacteria actually produce all these set of enzymes with which you can see these are the different targets of these particular enzymes with which the aminoglycosides are destroyed. So they have taken the pathway of offense, and by this offense they have destroyed the aminoglycosides applied to kill the bacteria. A very important role of a common antibiotic, a broad spectrum antibiotic, is to stop the protein synthesis. What they have done is they have modified the ribosomal structure. So sometimes what happens is the typical target of antibiotic is missing because the ribosome structure has been modified. So this bacteria can actually cause the mutation of the ribosome, and particularly this is true for a bacteria called vancomycin resistant Staphylococcus aureus, or VRSA, which has the unique gene VAN-A gene, and with the VAN-A protein this particular modification of the ribosome occurs and the bacteria is protected and they are not targeted by protein synthesis inhibitors. Okay? Now we come to the very important role of the transmission of the disease, how this disease is spreading. Obviously, one important role is through air, but it has been proved that this is a unique figure which shows that this person is having the Staphylococcus aureus in his body surface, and through contact it has been spreading to another person. So when we come with a contact with some infected person, we have every possibility that this particular Staphylococcus aureus will be coming into my body. When we talk about Staphylococcus aureus, there are two important strains. We call them as MRSA or methicillin resistant Staphylococcus aureus and VRSA or vancomycin resistant Staphylococcus aureus. Because these are the very recent antibiotics with which we try to combat these bacteria, but they have become resistant. So you can very well say they are the most powerful organism challenging human race in the present day world. And this particular MRSA can actually be transmitted through different parts of our body. It can include the nose, it can include the axilla, it can include the hand, it can include the groin, and it can even be present in the psoriatic lesion, apparently which can be autoimmune disorder, but they can harbor the different bacteria and spread from one person to other. And you can see in this particular figure, these are the nasal hairs and these orange bacteria, Staphylococcus aureus, adhering to these hairs. So very often when we sneeze, you can very well understand with the projectile, so many bacteria will be getting into another person. So perhaps self-hygiene is the best way to prevent this particular bacteria. Now, when we say that MRSA is there, definitely there must be some role or how to prevent MRSA. Now, there may be, when we say that, okay, methicillin is not acting, then we will try to change the antibiotic. It can be linezolid, it can be clindamycin, it can be tetracycline, even it can be trimethoprim or sulfamethoxazole, which is very commonly called co-trimexazole. But these are sulfur drugs, you know. So what I'm trying to say is, because one particular powerful drug is not acting in this bacteria, we'll try to take a host of other antibiotics and try to prevent that bacteria. This is the basic strategy of human that we adopt against Staphylococcus aureus. But when we come to this particular VRSA or vancomycin resistant, now this vancomycin resistant actually is resistant to vancomycin. Why? Because it has a thickened peptidoglycan layer, so the vancomycin cannot penetrate. And as a result of which, this particular vancomycin resistant bacteria can kill human being, and they have the unique VAN-A gene from which this particular modification of the bacterial wall occurs, so that they become resistant to vancomycin. So how to combat this? Basically, when we say it is vancomycin resistant, no other possible beta-lactam antibiotic can act on it. So the best way is to prevent the disease instead of combating the disease. So what we do is maintenance of personal hygiene. Very commonly we will use some soap solution to clean our hand at every moment, particularly those who are engaged in hospitals or those who are the nurses basically. Apart from that, there has been record that this bacteria can be transmitted through sexual intercourse. So safe sex is another method by which we can prevent this particular bacterial infection into the reproductive tract. As I was telling you, there are very common sanitizers, common chemicals with which we can use. That means we clean our hand and thereby the transmission of the bacteria can be reduced. One such compound is chlorhexidine. This is its structure of chlorhexidine, and it is used actually to apply osmotic stress on the bacteria, by which the bacteria will be killed and this bacterial transmission will not occur. Apart from that, there is another important compound which is the mucoporicin. The mucoporicin, this is the structure. This is an ointment which is given in the nasal ointment, which is a surface killer. And thereby those particular bacteria present in the nasal hair, as I was telling like some time back, will be killed. And there may be some particular amino acids which are also seen to be killing the vancomycin resistant bacteria. One such amino acid is L-homoarginine. Now, the best way to control in the present day may be biological control. And how this biological control? So there are certain strains of bacteria like the Staphylococcus epidermidis. What we can see here, this is the colony of Staphylococcus aureus. This is the colony of Staphylococcus epidermidis. You can clearly see one is antagonizing the other. You can see very well, this is the colony of Staphylococcus aureus growing so elaborately, but when they are grown along with Staphylococcus epidermidis, the growth is restricted. So that means they must be producing some bacteriocin or bacteriocin with which they prevent the growth of this dreadful Staphylococcus aureus. So you can see that if we have this Staphylococcus epidermidis, they look the same, almost the same. But this bacteria colonizing in the nasal tract will not allow this bacteria to grow, that is Staphylococcus aureus to grow, and thereby our body burden of Staphylococcus aureus may be reduced. So in this particular presentation, what we have learned the Staphylococcus aureus structure, how it was discovered, the different ways of its pathogenicity, and the different reaction that it brings about in human body. And there are different drugs with which we combat the disease, and at the same time we also discussed the different ways of these bacteria by which they can destroy the antibiotic and survive in human host. Okay, thank you.